In this lecture, we'll be describing the process of translation. Returning to the central dogma of molecular biology, remember that translation is the process by which RNA templates are converted to protein. This is the final step in converting a genotype, or a person's gene structure, to a phenotype, its appearance and function. Now, in translation, we have to convert a nucleotide code into a protein code. That is a polymer of nucleotides into a polymer of proteins. And this is a little bit different process than what we saw in replication or in transcription, where you're converting a nucleotide form into another nucleotide form of structure. And so there's a special way that we have to do this, and we call this decoding or translating the mRNA. Now, mRNAs encode proteins via a 3-letter nucleotide code. And each 3 nucleotides, or words we can call them, is what we call a codon. Now, there are 64 possible combinations of 3 nucleotides. You figure that out if you do the math. 4 to the 3rd power is 64. Of these, 61 of these codons, 3 nucleotide sequences, encode an amino acid. But we know that there are only 20 amino acids, so obviously there must be more than one codon for each amino acid, and that's absolutely true, as you'll see in the next slide. Now, the other 3 codons encode for stop codons. This is UAA, UAG, and UGA. These are codons which signal the translation machinery to stop adding amino acids. Now, there's another specialized codon, which is AUG. This encodes methionine, which you can find in any place in a protein. But it's also the signal to start translation, and as a consequence is always the first amino acid of any protein. Here's the code. This is the code by which codons in mRNA are converted to amino acids. Now, you can see in this that each 3-letter codon has a corresponding amino acid. Now, for the purposes of the USMLE, it's not important that you memorize this codon table. It is important, however, that you memorize the codons that we have highlighted. UAA, UAG, and UGA, which are the stop codons, and AUG, which encodes methionine and is also the start codon. It's critical that you know those to be able to answer questions on the USMLE. Now, here's an example of translation. Look at this and refer to your codon table and tell me what protein sequence does this RNA sequence encode. AUG, CAU, UUC, GGC, AGA, UGA. If you use the codon table to translate this protein, you'll find that this encodes methionine, histidine, phenylalanine, glycine, arginine, and a stop codon. If this were really being translated, you could say that the translation would start with a methionine and end with a stop codon. And what's the actual mechanism by which we decode an mRNA sequence to form a protein, the process of translation? This process involves a number of different components, one of which is tRNA. That stands for transfer RNA. Now, there's a tRNA which corresponds to each of the 61 protein coding codons that we described just a minute ago. Each of these tRNAs contains within its structure what we call an anticodon. This is a sequence of 3 nucleotides which is antiparallel and complementary to a particular codon. For example, the anticodon for the methionine codon, AUG, is CAU. That's antiparallel and complementary to AUG and allows the tRNA to bind to the AUG codon on the mRNA which is being translated. At the other end of the tRNA, as you see in the schematic below, the tRNA is charged with the amino acid that corresponds to the codon it binds. So in this tRNA, which we've shown here, the CAU anticodon, which binds to a methionine codon, contains on the other end a methionine tRNA. That is, the tRNA is bound to the amino acid methionine. So the codon which the anticodon binds should correspond exactly with the protein which it codes for. In this way, it's able to base pair with the codon and deliver the proper amino acids, effectively decoding the sequence. Now, the whole process of translation is catalyzed by a large protein ribosomal RNA complex, or rRNA complex, called the ribosome. In eukaryotes, there are two subunits to this ribosome. The 60S subunit is responsible for binding the aminoacyl tRNA, that is the tRNA which is charged with its appropriate amino acid. And then the 40S subunit is responsible for binding to the mRNA, the messenger RNA. These two come together and allow for the process of translation. Similar to transcription, translation involves four steps. There's a binding step, an initiation step, an elongation step, and a termination step. As in all other processes that we've described in molecular biology so far, this translation always proceeds in a 5' to 3' direction on the mRNA. The binding step occurs when the 40S initiation complex is formed. The 40S initiation complex consists of a 40S ribosome, a methionine tRNA, that is a tRNA corresponding to methionine with the methionine attached, some different proteins that we call initiation factors, the details of which you don't need to understand, and GTP, which provides the energy for this step to occur. This complex binds the 7-methylguanine cap that's found on the 5' end of mRNA and moves then in the 3' direction until that methionine tRNA finds the first AUG codon and its anticodon binds to it. Here's a schematic of that binding step. Here we have the complex being formed, the pre-initiation complex forming where the 7-methylguanine and the 40S ribosome bind. Here's the tRNA then binding to the appropriate AUG, and then we move on to the next step. Now that AUG codon base pairs with the anticodon of the methionine tRNA, and then the larger 60S ribosome binds the complex. Now the 60S ribosome orients the tRNA in one of three sites, and we refer to these sites as the P site, the A site, and the E site. The P site is where the tRNA attached to the growing polypeptide chain resides. The A site is where the next charged amino acid tRNA comes in, which corresponds to the next codon in the sequence. And the E site is where the tRNAs leave after their amino acid has already been added to the polypeptide chain. Now you can see this in this schematic again. Here's the 60S ribosome after it's bound to the 40S ribosomal subunit, and here's the P site, and here's the A site. And from this then we'll start initiating and elongating translation. Now initiation begins when the tRNA corresponding to the next codon, this is the codon which is immediately 3' to the AUG codon, comes in and occupies the A position on the 60S ribosomal subunit. The 60S ribosomal subunit then has an enzyme activity called ribosomal peptidyltransferase, and this catalyzes the formation of a peptide bond between the initiator methionine, that first methionine, and the second amino acid. The tRNA occupying the A position is now attached to a dipeptide, a methionine, and whatever the second amino acid is. The methionine tRNA is no longer attached to any amino acid now. So here's an example of how that happens. So here's the methionine tRNA bound in the P position. Here comes the next amino acid, which occupies the A position. This light blue box represents the peptidyltransferase activity, which binds the methionine to that second amino acid via a peptide bond. You can see now that the methionine tRNA now is no longer attached to a protein, and there's a dipeptide which is attached to that second tRNA. Now this process has to repeat itself over and over and over again in order for us to make a long polypeptide from the mRNA. The next step in this process is elongation. The ribosome has to move now down the mRNA, down to the next codon. So the ribosome translocates one codon in the 3' direction, 3 nucleotides in the 3' direction. The second tRNA, that now has the attached growing polypeptide chain, now occupies the P position. The methionine tRNA occupies the E position prior to leaving the ribosome empty-handed without an amino acid attached. Now the A position is vacant, and a third tRNA, the one that corresponds to the third codon in the mRNA, now comes in to occupy that position. Its detached amino acid forms a peptide bond to the growing peptide chain, via the peptidyltransferase activity, and this process repeats itself over and over again. Now you can see that here in the schematic. You can see here that the ribosome has translocated 3 nucleotides in the 3' direction, leaving the methionine tRNA, which is empty now, in the E position, the second tRNA attached to the growing polypeptide chain in the P position, and an empty A position. The next tRNA, corresponding to the third codon, then comes in and occupies the A position. The peptidyltransferase forms a peptide bond between the second and third amino acids, resulting in this structure here, where the second tRNA, which occupies the P position, now is no longer attached to an amino acid, and the third tRNA is attached to a tripeptide, a growing polypeptide chain. What happens then is the methionine tRNA leaves the E position, the ribosome translocates 3 nucleotides in the 3' direction, and the process repeats itself. The second tRNA now is in the E position, the third one attached to the growing polypeptide chain is in the P position, and the A position is vacant. The next tRNA will occupy that position, will form a peptide bond, translocation will occur again, and this process repeats itself over and over and over again, so that we translate now this mRNA sequence into a growing polypeptide chain. Now this elongation cycle will repeat itself over and over again until the ribosome reaches one of those 3 stop codons, which are UAG, UAA, and UGA. At that point, a protein releasing factor, which is a protein which recognizes that stop codon, comes in and occupies the A position. It binds the stop codon, binds the ribosome mRNA polypeptide complex, and it signals the release of the polypeptide from that final tRNA, again using the ribosomal peptidyltransferase activity. The whole complex then falls apart, dissociates, and what you end up with is a complete polypeptide chain, which has been translated from that mRNA sequence. Now this process requires a lot of energy. The energy comes in the form of ATP or GTP, and it's required at 6 different steps in the process. You require energy to charge the tRNAs, that is to attach the amino acids to the tRNAs. You require energy to bind for the binding step, where the tRNA initially binds with 40S ribosome. You require energy to initiate the complex. You require energy to elongate the process and form all those peptide bonds. Energy is required for the translocation from one codon to the next, and energy is required for termination of translation. Now, where this translation occurs within the cell, that is the site of translation, depends upon the kind of protein that you're making. It depends upon where that protein is supposed to go. Proteins that are destined for the cytosol, or for the nucleus, are translated in the cytoplasm on free ribosomes. But proteins that are destined to be secreted outside the cell, or proteins that occupy a transmembrane location in the cell, are translated by ribosomes attached to the endoplasmic reticulum. Now there's a clinical correlation to here as well. Just like the processes of replication and transcription, translation is a critical function for cells. If the cells are unable to make proteins, they die. And we're able to utilize this fact to kill cells that we don't want around, particularly bacterial cells. Now the important thing about bacteria that's different from eukaryotes is they have a slightly different ribosomal structure. Bacteria have a 30S ribosome and a 50S ribosome instead of the normal 40S and 60S ribosomes that we see in eukaryotes. Consequently, there are compounds, which we call antibiotics, which are able to bind the bacterial ribosome and halt protein translation and result in the death of these bacterial cells without interacting with the 40S and 60S ribosomal subunits in the human host. And so we can take these drugs which block protein translation and lead to the death of bacteria without affecting the protein translation occurring in our own cells. Some examples of some of these antibiotics are things like streptomycin, which affects initiation and elongation on the 30S subunit of prokaryotes, neomycin, which affects the whole process of translation, tetracycline, which blocks the binding of the aminoacyl tRNA to ribosomes, puromycin, which affects peptide transfer, erythromycin, which affects translocation, the movement of the ribosome down the mRNA, cyclohexamide, which blocks elongation, fusidic acid, which also blocks translocation, and ricin, which is actually a poison, affects multiple steps in protein synthesis. At this stage in the lecture, it's appropriate to talk about another clinical correlation, and this has to do with collagen vascular diseases. In this lecture and in the previous lectures, we've talked about DNA. We've talked about proteins that are involved in replication, transcription, and translation. Now, these proteins are found in every cell in your body and are typically not recognized by the immune system because they're normal residents. But if something happens, something goes wrong with the immune system, that it starts to produce antibodies against these particular proteins, what you often see is what we would commonly refer to as collagen vascular diseases. For example, antibodies that are raised against DNA, particularly double-stranded DNA, are often hallmarks of the disease lupus or systemic lupus erythematosus. Antibodies which are raised against histones, these are the proteins, remember, that package DNA in the cells, often lead to drug-induced lupus or a lupus syndrome that's induced by taking particular medications. There are also antibodies which are raised against ribonucleoproteins. Remember, the ribonucleoproteins are things either in the ribosome or in the spliceosome responsible for mRNA splicing. There's a particular ribonucleoprotein called Smith. The Smith antigen, an antibody raised against the Smith antigen, also is involved in the pathogenesis of lupus. One raised against the U1 ribonucleoprotein causes mixed connective tissue disease. And antibodies raised against the nucleoproteins SSA or SSB, which are also called Rho and Law, result in Sjogren's syndrome. Finally, remember we talked about topoisomerase I, which is responsible for unwinding DNA and DNA replication. An antibody raised against topoisomerase I called SCL70 is responsible for the disease systemic sclerosis. This concludes our discussion of translation and concludes the part of the molecular biology section of this lecture.